So for today's Zoom meeting, the first thing that we'll do is jump back into this PowerPoint that we were talking about on Monday. And I said, hey, make sure you go read chapter 8 because I knew you were wrapping up the test and go through the PowerPoint, but I wanted to come back in and talk about the Mendelian examples, well, the non-Mendelian examples, I should say. So basically, I'm jumping in at slide 38, a little bit ahead of that image. And just to recap, genotype is what proteins the genes code for, and phenotype is what is actually expressed. And it's a combination of what your genes code for and how the environment influences gene expression. So you could have genes that code for, you know, the ability to grow tall, but if you don't get enough to eat, you won't be as tall as your genes code for. That sort of thing. Or you could have a gene that will code for something, but there are other environmental interactions that impact how those genes are expressed. And I have an example of flower petal color and hydrangeas in this PowerPoint presentation. The other thing that's going on is, remember that Mendel chose specifically 7 characters or heritable traits that had an either or, purple or white, flower color, tall or short plants, flowers that were growing along the sides or in the terminal position at the top. And he avoided and ignored all of the other characters, the things expressed by pea plants, that looked like maybe they were mixed or blended or he couldn't sort them out. And one of the things that we now know that he didn't was that some of those characters were on the same chromosome, and we know that on a given eukaryotic chromosome there are many, if not hundreds, of genes that are coded for. And basically his principles of segregation and independent assortment apply to more complex patterns of inheritance, but there are layers of detail that don't make it as cut and dry as dominant and recessive alleles. And there are some genes for which there are more than 2 alternatives. Imagine if you wanted ice cream and your only 2 options were chocolate or strawberry. Two alternate versions. Well, we know that there's chocolate and strawberry and vanilla and pineapple and, you know, everything in between. And so there's multiple flavors of ice cream. Well, there can be multiple alleles for a given gene. One of the examples is blood type, and we'll get to that in a little bit. Okay, so when alleles are not completely dominant or recessive, and they might be expressed at the same time, if one gene doesn't turn off the other, the gene doesn't go away. And so what we're calling recessive doesn't mean that that gene or protein isn't being expressed. It just means when something is dominant, that version of the allele is the one that we see in the phenotype. Well, some genes that have, you know, 2 alleles are both being expressed, and we can see the expression of both. And sometimes we see a layering or a synthesis of those 2 genes working together being expressed. Sometimes we see that there are many genes that affect a given phenotype, not just a single gene. And so we have to consider all of those scenarios. Okay, so the first slide gives 4 examples that are not strictly Mendelian. So the top one is the one Mendel came up with, and he coined the terms dominant and recessive. But what happens if the heterozygote, the one that has 1 copy of each allele, is an intermediate in between the fully homozygous dominant and the fully homozygous recessive genotype and phenotype? We call that incomplete dominance. And it means that it's sort of like a blending, where both proteins are made and you see something that is neither one or the other. So when it's neither one or the other, we call it dominant. And that's different than codominance, where the heterozygote expresses both phenotypes from the two respective genes or versions of the gene, and they're both expressed fully and separately. So incomplete dominance looks like blending, and codominance is double expression, full expression of both of the alleles. And blood type is a great example of codominance. Blood type also happens to be an example of multiple alleles, because you probably know that you could be an A blood type, a B blood type, an O blood type, or an AB blood type. But you're not ABO. You don't get all three. You're either AA, BB, OO, or AB. Or if you have one allele that codes for A and one that codes for O, we would still call you type O blood. And I'm going to get into that in a couple of minutes here. And the last one on this slide is pleiotropy, and that's where there's one gene, but it's not expressed in just a single way in an organism. It's expressed at a molecular level, at a cellular level, at a tissue level, at an organism level, so that there are multiple ways in which the protein impacts the way that the organism functions. Yes, you may get popsicles to share. My daughter's having a summertime day outside. Okay, so the other two examples that fit into these couple of slides are epistasis and polygenic inheritance. And the example I have for epistasis is for fur color in mice. And basically, the one gene, depending on which alleles the mice have, one gene says whether the mice is going to get brown fur or black fur, but a second gene says whether or not the fur gene is going to be turned on or off. And so if you have a mouse that has black fur color coded for, but the other gene says that first gene is turned off, then you get a white mouse. And so one allele affecting the other is what you call epistasis. And then polygenic inheritance is when you have a phenotype that depends on multiple proteins being produced by multiple genes, poly being many. And there are an awful lot of human genes and genes in other organisms that are polygenic, and there's some great examples besides skin color that are in this presentation. All right, so the example given here with incomplete dominance, do you see how the true breeding parents have red or white petal color? And when we crossbreed them together, the offspring, the F1 generation, are all pink, and that's because it's a heterozygote. So the heterozygote shows incomplete dominance that's neither the exact expression of the protein coded for by the gene of one or the other parent. It's a combination of the two blended together. So this color here is in snapdragons is really evident of that. Then let's look at blood type here. Do you see how if you are, and the way we write this is a little bit different as opposed to just a capital letter being dominant and a lowercase letter being recessive. We actually use superscripts to explain the genotype. And so to be type A blood, which is what the phenotype would be described as if you got your blood drawn and you asked what your blood type was. If you are type A, you could be IAIA, this that you see right here, or you could be IAI. And the two little I's, if you look down below, that's when you're type O. And what this really means is that if you are type A blood, then you have the allele that codes for a specific protein A to be on the surface of the red blood cell. And if you are type B, you have the allele that codes for the type B protein to be on the surface of your red blood cells. And if you are AB, it's called codominance because you fully express the A protein and you fully express the B protein separately and together, not blended, but separately and together, okay? If you are type O blood, what do you code for? What's not on the surface of that red blood cell? There's no recognition proteins. And so type O is the absence of those recognition proteins. So why would a person with type O be a universal donor to A, B, and A? Because if I were to give type A blood, type B blood, or type AB blood to an O person, they would reject it. They would have an immune response that would cause them to reject it. But I can give a person with AB blood, type A or B or O, and have it not be rejected. And who can I give the O blood to? I can give it to O, to AB, to B, and to A. And so that's with the protein on the surface of the red blood cell. You've probably heard of Rh factors, whether you're like A positive or A negative. That's a different protein and something else that has to do with blood type. So actually the universal donor is O negative. And my husband and I both have type O blood. And we were a little bit surprised to find out that one of our kids is O negative. So all of our kids are O. And so we just assumed all of our kids will be O. And that's correct, but that's one particular gene. The other gene that comes into play when we talk about blood type is the Rh factor. And so we have a kiddo who is O negative. And the reason that they got negative is we must both have a recessive negative Rh factor allele. So blood type itself, A, B, AB, or O, that's multiple alleles. The Rh factor is regular dominant and recessive Mendelian inheritance. And remember, it's also multiple alleles because there are 3 different alleles that we're looking at, A, B, or O. Okay, so this slide talks about what happens when a dominant allele does not subdue a recessive allele and the alleles don't interact. That is the exact definition of what I was showing you before here. So when we talk about codominance, what it really means, literally, is that the dominant allele doesn't subdue a recessive allele and the alleles don't interact, so that both gene versions, both alleles, are expressed, okay? So sometimes it really depends on the level at which we examine a phenotype. When we look at Tay-Sachs disease, which is not a good genetic disease to inherit. If you have the allele that gives you the disease, then it produces a dysfunctional enzyme that can't break down lipids in the brain. And so you end up with an accumulation of lipids in the brain, which causes the symptoms of the disease. And at the organismal level, so when we look at the whole organism and what the genome codes for, the allele is recessive. So that would mean that we're looking for, you know, we don't want to see homozygous recessive individuals, because then they're going to have 2 bad copies of the protein, of this enzyme. At the biochemical level, the phenotype is what we call incompletely dominant. And that means if you're a heterozygote, that you have some good enzymes and some bad enzymes, right? Meaning that you could remove some lipids from your brain, but you might still express Tay-Sachs disease, but to a more moderate level because you only have 1 copy. And when we look at that even further, we say at the molecular level, the alleles are codominant, and that's because one good enzyme doesn't turn off one bad enzyme. They're both produced. And so Tay-Sachs disease is just a really interesting way to look at all these, you know, non-Mendelian genetics patterns. Okay, so I promised we'd talk about pleiotropy. So when we have multiple phenotypic effects, some really common examples for humans are the diseases or illnesses, sickle cell anemia and cystic fibrosis. So when we look at sickle cell anemia at the molecular level, when you have a faulty gene, you have hemoglobin that needs to capture oxygen. And when the hemoglobin protein is misshapen based on just one amino acid substitution, the shape of the folded hemoglobin changes and it's not as able to bind to oxygen. So we have a molecular structure of the protein that's altered, and we have a change in function where it's not as able to bond to oxygen. And then what you see on a tissue level is that the red blood cells are kind of funny shaped, and that's why this disease is called sickle cell, because they're curved and they're much more rigid, and those blood cells are more likely to break. And so you see at the tissue level, you see a funny shape, you see an inability to hold on to oxygen, and you see blood cells that get trapped in the arteries. And then on the organ level, then you actually see problems with oxygenation and tissue damage and death. And basically, if cells don't get enough oxygen, then they can't perform enough muscle contraction and activity because they're short on ATP. And so pleiotropy is evident here because you're seeing the effects of the the bad allele at the molecular level, at the cellular level, at the tissue level, and then what do we see at the organismal level? You see anemia, you see shortness of breath, you see weakness and fatigue and exhaustion, and an impact on the ability to live life normally. And cystic fibrosis is similar. There are multiple problems also where we are looking at this inability to move chloride ions across plasma membranes. And that may not seem like such a big deal, but if you can't move salts properly, then it's hard to have appropriate neurological function. And then what happens with having salts be the wrong way is, remember that water follows solute. And so if you can't move your ions properly, then you have fluid buildup. And one of the things that we see with cystic fibrosis is mucus buildup in internal organs, and then funny absorption in the small intestines. And that's all based on, an inability to move the ions where they need to be. So I'm going to skip past these couple of slides here. All right, so I already explained the mouse one, and I've got an example in another slide presentation with some questions that we'll go over with that. So I'm going to go past this one. And then here's my example of polygenic inheritance, where we're looking at two individuals who, and we're seeing that they're heterozygous for all three of the genes that give the skin pigmentation phenotype. And so they all have an intermediate skin tone. And what happens when they have children is every time they have a kid, we have a pretty significant chance for this kid to get an intermediate skin tone, either just like the parents, or a little bit lighter, or a little bit darker. And it's totally within the realm of expected phenotypes to have that happen. And then you can also see that there's a chance for an offspring to get all proteins that code for minimal or absence of pigmentation. And then it's also possible for an offspring to have the chance to get all of the alleles that code for the maximum amount of pigmentation. So it's really interesting to see that that come out in a family where the phenotype of skin color isn't quite what you would expect. And really, it just is a great illustration that all humans is the combination of you know, our genotypes being expressed, given to us by our parents, but that more than anything, we're all expressing human genes, human DNA. And so we should remember that first, that we're all humans together. And you can see in this particular slide that this is a bell curve, and where that comes from is the majority of the phenotypes from those heterozygous parents fit in this intermediate range, but you will see some that are lighter than the parents and some that are darker than the parents, and it's totally normal genotype expression. And we just say, hey, there are many genes in charge of pigmentation, and so that's polygenic. Other polygenic traits include, body type. So your ability to, you know, build muscle or not, the shape of your body, how tall you are, those all come from your parents, and not just from one set of genes, but from multiple genes interacting together. Hair color is not just one gene, it's multiple. And do you remember a week ago I said eye color wasn't strictly either or? There's more than one gene that codes for eye color, which is part of the reason why I have hazel eyes, even though my father has blue eyes and my mother has brown eyes. There's more than one gene behind the scenes controlling eye color. And so the bell curve is something that is shows you that polygenic traits are real. And the interesting thing is that the individual genes of a polygenic trait follow Mendel's laws, but together they don't produce any kind of 3-to-1 or 9-to-3 to 3-to-1 ratio. And it's because we're looking at their additive combined effect on the phenotype. And that seems to be distributed across a bell curve, where the extreme or rare phenotypes are, you know, the left and right ends of the bell curve, but most individuals fit. It's quite possible to see a bell curve shift in one direction or the other, or even split into two dips, to a dip and two hills. And if you think about like human birth weight, one of the things that's happened with survival of infants, you know, with hospitals, with medical care, with nutrition, is we've seen a shift. We've seen a shift in this curve where if the x-axis is birth weight and the y-axis is the number of individuals, we've seen a shift to the right where the majority of babies are a little bit heavier at birth, at full term, than babies were, you know, 100 years ago. But we've also seen a bit of the curve like flattening out, where we see more babies than 100 years ago that are heavier, and more babies than 100 years ago that are lighter. Think about what intensive care units for infants has done to improve the survival of babies who are born early, or who are born in, you know, this time period where 100 years ago they wouldn't have survived. And that's based on technology and you know, maternal nutrition. And so we see a shift in the curve. And height, has anybody ever seen a bed from a couple hundred years ago? People were shorter. So height is an additive polygenic trait, but also we've seen natural selection, evolution towards individuals being taller over time. So just something cool to think about. I don't know of any instances where eye color has shifted one way or the other. That would be sort of fun to take a look at. Okay, so here is my example. Hydrangea flowers might have the exact same genotype, the exact same alleles, but their phenotype may be different, and that's based on the pH of the soil that the plant grows in, whether you get pink flowers or blue flowers. So the environment definitely affects genotype expression. And remember, we call genotype expression the phenotype. Okay. So why did Mendel study peas and not people? Well, peas are much more cooperative compared to humans. The generation time, like how long it takes to grow a pea plant, get the flowers, get the seed pods produced, and then grow a new set of plants is pretty quick compared to how long it takes humans to be born, grow up, and produce offspring. Pea plants make several hundred pea seeds, and parents, generally make one baby at a time if you're human, right? And also, nobody really cares if you crossbreed pea plants, but humans aren't really going to follow those kinds of breeding directions. So it's important when we look at genetics and patterns of inheritance that, when we study things, we're looking at species that it's okay to, manipulate and do experiments on. So how do we do this with humans? We did, in the past, pedigree analysis, where we looked at what your ancestors had. Now we do a lot of genetic testing, where we can see what genes people have and look at things like that. One thing that I haven't talked about yet is sex-linked traits. And sex-linked means that the genes that are giving a particular phenotype are on chromosome 23, either on the X chromosome or on the Y chromosome. And there are many more sex-linked traits that are X-linked than Y-linked, and that's because the Y chromosome is tiny and the X chromosome is bigger, and so there are more genes coded for on the X chromosome than on the Y chromosome. And also, one of the things that you see is that if there's a particular trait, you often see sex-linked traits show up in males more often. And why is that? Because if it's a recessive thing and the male has it, well, the male only has one X, so it's going to be expressed, where a female has two X's. And if it's a recessive trait and she has one dominant X and one recessive X, then you don't see that sex-linked trait being expressed. So X-linked recessive traits are traits that result from a recessive allele on the X chromosome, and a woman would have to have two recessive X's, two genes that were recessive for that trait, one on each X, in order to show that. And there are about a little more than 100 different human conditions that are caused by recessive alleles found on X chromosomes. And basically, you see those X-linked traits expressed in males, which may seem kind of counterintuitive because X means woman, right? No, 2X means that the sex is female, and XY means that the sex of the individual is male. Okay, X-linked dominant, there are a few of those, and again, if you're the male and you have the dominant allele, you're going to express it, but also in females, you just need one. Okay, so here's my example of pedigree analysis. And if you were to look at a pedigree chart, when the symbol is a square, it means a male individual. When it's a circle, it means female. And then if there's a horizontal line between two individuals, it means that they had kids together. And then down beneath that, you would see from left to right, their offspring. And if the circle is not filled in, then it means they don't express the phenotype. If the circle or the square is filled in, then they do. And so what we're going to look at is ears. And so you could all check your ears. Are your ears lobes, are they attached as on the left here, or are they detached as on the right, free swinging earlobes? And you inherited that from your parents. And looking at this pedigree chart, you can see that attached earlobes is a homozygous recessive trait. And so when we look at this person here on the right, how do we know if they're homozygous dominant or a heterozygote? And we can look back at their parentage, and you can see here, here's their mom, here's their dad. And how did this person get a sibling that had attached earlobes? Well, that must mean that both of their parents were heterozygotes. If we know that their parents had unattached or detached earlobes, but they have a child with attached earlobes, then we know that both the parents had to be heterozygotes here. And you can see that the mom has a sister, so these women have an aunt here who has attached earlobes, who's homozygous recessive. And the dad, they have uncles on that side who also have attached earlobes. So you can see that it's not that all members of a family are one way or the other. The only way it's going to be all attached is if everybody is homozygous recessive, and the only way it's going to be all detached is if everybody in the generations is homozygous dominant. And if we look back, we actually can't tell based on the parents whether this person is homozygous dominant or heterozygote. We know they have to be one or the other. How would we find out? Well, it would depend on who this person with detached earlobes mated with and what their offspring were like. So if they had 10 kids, if she was to mate with somebody who had detached earlobes, and that person had a history of siblings with attached earlobes, or had parents with attached earlobes, then we would know that the mate was a heterozygote. So if the mate's a heterozygote and she's this, then at some point we would expect that they would have some offspring with attached earlobes. But if they have 10 kids and you never see attached earlobes, what would that indicate about this person's genotype without having to do a gene test? It would indicate that she was homozygous dominant. And so you have to do the test cross with somebody who is homozygous recessive. And that's what I was talking about with the flower petal colors the other day with Mendel's experiments. But how easy is it to tell a human, okay, go marry that guy and have kids with him because he has detached earlobes? It doesn't work, right? So we look at pedigree charts and we infer, and then also more recently we do genetic testing to look at specific genes of interest. Okay, this example just looks at, it's a reminder of Mendelian genetics and looking at homozygous recessive and homozygous dominant. And I just want to walk you through segregation again, the law of segregation. And if you look at the parents here, the kids are shown, but the parents are shown that they both have normal pigmentation. And so this is a gene that codes for whether or not the other genes that impact skin pigments are turned on or turned off. So we're talking about epistasis here, a gene that determines whether those other skin color genes are on or off. And both of the parents demonstrate normal pigmentation, and looking at their genes, we can see that they're heterozygotes. And that means, well, what can they give to their kids? We're looking at, all right, they're alternate forms of the allele. When the gametes are formed, each gamete has one version of the gene or the other, never both, okay? So those genes, those alleles separate into the gametes. And then at fertilization, we have either, we basically have a 25% chance that either one of these combinations would occur because of what happens during meiosis 1, okay? And so for these parents, what is the chance each time they have a child that they will have a child without the ability to have skin pigmentation? It's a 1 in 4. So they have a 25% chance that each child that they have would be what we refer to as albino, meaning the absence of skin pigmentation, okay? And I'm trying to escape out of the slideshow here. I'm just going to walk you back and remind you here that when we are looking at the law of segregation, the first concept is that there are alternative versions of genes. So you saw that, the big A or the little a. The second concept is that an organism inherits 2 alleles, one from each parent. So you saw that an egg carried 1 allele and the sperm carried the other. The third concept is that if they're different, if the alleles are different, then one determines the organism's appearance, so one of the alleles. And so in the case here, if you had a big A, that would determine that you had skin pigmentation, whether you had 1 big A allele or you had 2 copies, okay? And then the fourth concept is basically explaining that the 2 alleles separate during gamete formation and end up in different gametes. So all of that is part of the law of segregation. Okay, so I'm gonna move this out of the way. Box is in my way. All right. I want to make this big here and just walk you through a couple of examples, okay? All right, so this is not in Canvas, it's just an extra file that I have to walk you through. So this question says, what is not true about alleles? So if we look through these alternatives, the 3 of these will be correct and 1 of them will not be. So looking at A, 1 allele is from each homologous chromosome. So if you think about where do the alleles go in gametes? Well, 1 comes from each of the homologous chromosome pair. And 1 of those alleles is from your mother and 1 is from your father. That's correct, which makes sense that then 1 allele would come from the sperm and 1 would be from the egg. Now remember me talking about 6 fingers versus 5 fingers and how most of us are 5-fingered and that is a homozygous recessive trait? Well, that means that we inherited a 5-fingered allele from our mom and a 5-fingered allele from our dad. And those alleles are not different. So see how D says different alleles are always found on homologous chromosomes? They might be if somebody is a heterozygote, but for any individual that is homozygous dominant or homozygous recessive, those are the same alleles found on the homologous chromosome set. So A, B, and C are correct, but D is false. All right, number 2 asks, how can one gene locus contain different information for a given trait? So remember, the locus is the place on the chromosome where the allele is, where the gene is coded for. So which one of these is going to be correct? So 3 are false and 1 is correct. So A, 1 allele can represent different traits. Well, a trait is something that is coded for by alleles. So this is putting that in reverse and basically saying one slice from a pie can represent many pies, and that's not accurate. B, 1 trait can represent different alleles. Well, what do you think about that one? That one is maybe a little bit complicated here. But what is a trait? It means it's a particular set of it's a gene where a set of alleles is being expressed, but it still sounds kind of kind of funny. So one trait can represent different alleles. Well, whatever the trait is, it's a couple of alleles and that are at a particular locus, and it's the same gene that's being coded for. So let's see if a different answer makes more sense. So look at C. A variety of alleles for one trait can be represented at the same locus. So if we've got a gene that's coding for blood type, how many different alleles are there for that gene? We know we've got A, we've got B, and we've got O. And what can be coded for at that spot on the chromosome? At the locus, you can code for A, you can code for B, or you can code for nothing, which is O. And so there's a variety of alleles for one trait that can be represented at the same locus. So we know C is correct and makes sense. Now let's look at D. The locus is found at the same place but on different chromosomes. Well, a locus by definition is where a gene is located on a given chromosome. So whichever chromosome codes for eye color, those genes are found on the same chromosome every time, not on chromosome 2 in some individuals and chromosome 6 in others, but always on the same chromosome in the same spot. So C is correct here. Okay, number 3 says a plant that has 2 identical alleles at the same gene locus is said to be, and look at these, heterozygous, true breeding, self-fertilized, or cross-fertilized. Which of these is another way to say homozygous or the same?
True breeding?
True breeding, yes. Okay, so, and heterozygous is 2 different alleles at the same gene locus for a given trait. Okay, number 4, what can you conclude about the genotypes of 2 white-flowered plants that always produce white-flowered offspring? So the always here tells you that something is true breeding. And when we say something is true breeding, another way to say that is it that they're heterozygous or homozygous?
Homozygous.
Homozygous, yes. And so that eliminates A and B. And C and D, your choices now are these white flowered plants homozygous for white flowers or for purple flowers? What are they producing? White flowered offspring. So the version of the gene that they have is the allele for white flowers. And because they always make white flowered offspring, then we would know that they have 2 alleles, 2 versions of the gene that code for white. And because those 2 are the same, they're homozygous. Okay, number 5, when one individual is homozygous dominant, so that means 2 copies of an allele for a given gene that are the same and that are dominant, meaning that's the one you see in the phenotype. And another individual is heterozygous for the same trait. Then what would you see? Do they have the same genotype? Well, let's look at fingers, 6 fingers versus 5 fingers. Somebody is homozygous dominant is going to be big F, big F. And somebody who's heterozygous is big F, little f. They do not have the same genotype because one is big F, and the other is big F, little f. So A is wrong. Look at B. Do they have the same genotype? Sorry, A is not wrong, it's B that's wrong. B says they have the same genotype, and we just said, well, one who's homozygous is going to be big F, and the other one who's the heterozygote is big F, little f. So that's different genotypes, okay? Now, what did I say about 6 fingers? Whether you have 2 alleles for 6 fingers or 1 allele for 6 fingers, what do you have? If it's a dominant trait, whether you have 1 allele for 6 fingers or 2, what are you going to have? 6 fingers. That's what dominant means. Okay, so the 5 finger isn't expressed. The 6th one, the 6th fingered trait is what's expressed. So when you're looking at Mendelian genetics and we're looking at dominant versus recessive, the homozygous dominant individual and the heterozygous individual have different genotypes, but they have the same phenotype. They both express the dominant phenotype. And if I were to bring this one back in. Do you remember this slide from the other day? The phenotype, whether you're big P or big P little p, whether you're homozygous dominant or a heterozygote with one dominant allele, the phenotype is all the dominant phenotype, or purple. So it's the same for flower petal color as it is for fingers, okay? So C is incorrect. They're not true breeding because a heterozygous individual can't be true breeding. So let's go back to A. They have different genotypes, but what do they both express? The same phenotype. So A is correct. Okay, number 6. This is looking at eyelashes here. So assume that long eyelashes are dominant to short eyelashes. The heterozygous person with long eyelashes mates with a person with short eyelashes. So what would that, I want to insert a little box here. Okay, so here's my heterozygous person, L, and they mate with a person with short eyelashes. So if long eyelashes are dominant, then we know that the person with short eyelashes must be little l, okay? And what is the genotype ratio of their offspring? Well, if I were to give them a Punnett square, then we would see that, this is where it's tricky. If I want to draw one, I need to insert a table. That's what I need. There we go. So here we would have the gametes of the parent. Oh, I don't like that it's doing white.
Does it matter if we don't know what the person with short eyelashes has, or that's
you do know, because it says here that long eyelashes are dominant. So when it tells you that, when something's dominant, we use the capital letter to symbolize it. Okay, so if you're a heterozygous person with long eyelashes, the heterozygote means you have one of each. Okay, so you're big L, little l, but you have long eyelashes because long is dominant to short. And it says that they mate with a person with short eyelashes. So in order to have short eyelashes, you have to be little l. You can't have a dominant allele or you would have long eyelashes. So the first parent can give big L or little l, an allele that codes for long or an allele that codes for short. But what can the other parent give? Only little l, right? They can only give one, they don't have a choice. Their gametes are going to be identical. So you want to ask the question again?
I was talking to my kid.
Oh, okay. What's the question again?
Oh, I see. Oh, the question was, how do I know what the person with, what the mate has?
Okay, and it's given here. So there's,
yeah, I'm sorry.
The information about dominance and recessiveness is given here. And then you're told here, heterozygous person, and then you're told who they mate with. So these are the individuals.
Yes.
Genotypes. And the phenotype, if it'll let me in here, is long times short, right? Somebody with long is mating with somebody with short eyelashes, okay? But because of the law of segregation, we know that each of their gametes is haploid. So the person with short eyelashes can only give a short eyelash allele, and the person with long eyelashes gives one or the other. And so when they mate, their chances, if we combine their gametes together, these two make big L, little l. These two make big L, little l. And over here, this one and that one make little l, and this one makes little l, okay? So every time they have a kid, they have a 50% chance or a half chance that that child will have long eyelashes and a half chance that that child will have short eyelashes. So which of these matches the genotype ratio? C, right there. So you see how the phenotype and the genotype can be predicted by completing the Punnett square, where genotype is the allele combination and phenotype is describing the trait, long versus short, okay? And remember, it's expected. Is it possible that they could have all long eyelashed kids, even if they have 10 kids? Yes. The probability that every single one of their 10 kids would have long eyelashes is 1/2 to the 10th exponent. It's pretty rare, but that has to do with laws of probability. Okay, so the next one. Remember, I promised that you don't have to do a dihybrid cross, but you still need to understand what happens when you have individuals who are heterozygous for more than one trait. So this individual is big S, big Y, little y. And what would you actually see? So if you know the distributive property from math, and we have to look at, okay, their alleles would be big S or little s, and big Y or little y, right? But how do they combine? We can have big S with big Y or big S with little y, right? Because of independent assortment. If I were to put because these genes are not on the same chromosome, so what the gene that codes for S does, what that chromosome does, doesn't determine how the chromosomes that code for Y line up. And so we could end up with, and the distributive property is you take the first in this set, so big S times big Y, so SY here, and then you take the outer, so big S times the little y, and that's this one. Then you take the inner, so little s, big Y, and then you take the last, which is little s, little y. And that's how you figure out the gametes of the parent organism, because we can see big S or big S, and we can see little y or big Y, but we have to come up with the 4 different combinations. And so the only one, oh, and I had changed it from numbers to letters, and it looks like I forgot to delete the numbers here, and that's what's confusing. Sorry about that. So what's the only one that shows correct gametes? It's this one, okay? That shows all of those options together. So anytime you have an individual who is heterozygous for 2 traits, they can have 4 different types of gametes. And that's why when you look at a dihybrid cross, you would then see all of these gametes down the left side and then potentially down the right side if it was 2 individuals mating who were heterozygous for 2 different traits. But I'm not going to make you actually do the high dihybrid cross. I just want you to understand that this illustrates independent assortment, okay?
The hybrid cross actually, I mean, like feels easier for me than trying to, yeah, like Lindsay or somebody said, like the foil method.
The foil method, yeah, that's what that is. It's the distributive property, and my 5th grader gets it because we were using it to calculate area of rectangles where the area was given as a mixed number, like 5 1/4. And 5 1/4 times 2 1/2, and he at first wanted to just do 5 times 2 plus 1/4 times 1/2, and it means he's totally missing the the 5 times the partial and the 2 times the partial are a part of it. And so we had fun discussing the distributive problem. All right. So if on the test or if doing homework, something that it might help you, I'm not sure how quickly I can come up with it, but we'll see. We'll pull up our friend Google and we'll say dihybrid cross calculator. This didn't exist when I was in school, in college. It would have been kind of nice, but you had to know it and do it cold. But there's more than one of them. But if I click this open, you can actually put into this calculator the alleles and calculate things out. There are different calculators that exist. And so you could actually find one. Look, one is also just calling itself a cheat sheet. So if you were struggling with the homework, one of the things that I put into the homework assignment was a link to this website for Arizona's education website, where you can actually do a Punnett square, and this one is the dihybrid Punnett square, and it walks you through that one, and then there are many other problems that you can look at. So that one's pretty cool. And I put it right in the homework because I want you to understand how to use it, not agonize over the calculations, right? For either a monohybrid, single traits, or double traits if you were interested. Now, this one looks really tricky, but I'm going to show you how to figure it out. So it says a white forelock is a patch of white hair, typically at the hairline, and a cross produces 1,836 people with white forelocks and dimples, 612 people without white forelocks but with dimples, 597 people with white forelocks and without dimples, and 208 people without white forelocks and without dimples. And what are the genotypes of the parents? And you're like, what? How do I even figure that out? Well, I want you to look at the numbers are approximately 1,800 to 600 to 600 to 200. And what can you divide all of these by? 200. And what does that reduce to? 9 to 3 to 3 to 1. So what did I tell you? That if you get offspring with a ratio of 2 traits that are a 9 to 3 to 3 to 1 ratio, that it means both parents are heterozygous for both traits. Okay, and this still shows independent assortment and segregation of alleles. Okay, and I need to shrink my font size here so that you can actually see the results. And which one of these shows an individual heterozygous for both traits? C.
Yeah.
Okay, so I'm not asking you to do a crazy dihybrid thing. I'm asking you to recognize this about Mendelian genetics. Okay, that's what you're looking at. It's not going to get more complex than looking for that. And in fact, in my previous final exams, I've just said, what does 9 to 3 to 3 to 1 ratio actually mean? And then you have to say both parents are heterozygous for both traits. It shows independent assortment and segregation of alleles. Okay, so let's look at when we think about heterozygotes and we think about expression, what does that 9 to 3 to 3 to 1 ratio actually mean? Well, remember that the 9/16 is individuals with dominant phenotype for both traits, okay? And the 3/16, both of them, both sets, have the dominant phenotype for one trait, and it's going to alternate. And again, let me shrink my text. So this 3 out of the 16 options and this 3 out of the 16, because 9 plus 3 plus 3 plus 1 is 16, means that 3/16 are going to show dominance for one and recessiveness for the other. And then 3/16 are going to show dominance for the second and recessiveness for the first. And then lastly, you have 1/16 of the offspring expected to show the recessive phenotype for both traits. So let's look at this. If we look at the one that has the 9/16 ratio, we figure out which two traits, what the dominant version is. So if we have 1,836 people with white forelocks and dimples, what is dominant? White forelocks and dimples. And then look at 612 people without white forelocks and with dimples. So the absence of white forelocks, okay, they're showing recessive here. And then with dimples, they're showing dominant. 590 people with white forelocks, so they're showing dominant white forelocks, and without dimples, recessive for the second one. And 208 people, 1/16, without white forelocks and without dimples, so they're showing recessive. They're without the forelocks and without the dimples. So it says, what are the dominant traits? So what did we pick out? What produces the 9/16 ratio? It's this one right here. White forelocks and dimples. And so A would be your correct choice. So again, it's looking at that 9:3 to 3:1 ratio that Mendel came up with this dihybrid cross. So do you think you can remember the 9:3 to 3:1 ratio and what it means? I'll post this video so that you'll have it, okay? If you wanted to go back through. Number 10, this is getting at the 23rd chromosome and what happens. What's the probability that a newborn child will be male? Well, what is the mom? XX. And what is the dad? XY. What does the Punnett square look like? I'm just going to grab this guy here. So what happens to the sex gametes? Here's mom. Here's dad. And what do we fill in? What are their kids? We have a 50% chance that they have what? Oops. There's a 50% chance that they'll have a girl every time and a 50% chance that they will have a boy. So what's the probability that any given newborn child will be a male? How many of these boxes are filled in that show a male phenotype and genotype?
50%.
50%, that's right. Now, here's a question for you. What's the probability that 2 children in a row will be male? Okay. So the first kid, it's a half. Okay, so here, probability, if I could spell it, 2 kids in a row are male. So the probability of the first kid and the probability of the second kid are both a half, right? To be male. What's their cumulative probability?
25.
Yeah, the cumulative probability. Oh, come on. Is multiplicative. Say that really fast. But all that means is it's a half times a half, which equals a fourth. Okay? Okay, number 11, how many do we have? Oh, there's a lot of them. So what is the genotype of an X-linked male with a recessive disorder? Okay, so this is looking at what did the slide show, the presentation show we're supposed to do with that. So a male is XY, okay? And if he's recessive, then he shows, it's gonna be a small letter. Which one of these shows XY with a small letter?
C.
C, right? X, this one right here, okay? So you got that. Give me 1 second here. All right, in the meantime, we'll do the next one. So this should be familiar. Snapdragons were those flowers that I was talking about where you have a true breeding parent that's red and a true breeding parent that's white. And then when they breed together, they have heterozygous offspring that is pink. So what do we call that type of inheritance?
B.
Mendelian genetics is complete. This doesn't mention sex chromosomes at all, so that takes away A and D. So you have to decide, is it incomplete or is it codominance? Which one of them is when the phenotype appears to be a blending of the alleles of the two parents?
B.
B, that's correct. And codominance is when they're both expressed separately, but they're both present. Number 14, hemophilia is an example of an X-linked recessive disorder. So just take a minute, think through this one, and I want you to try to work it out.
I thought only males got it.
No.
She has 2. I don't know what subscript to give it.
Okay, so
2 lowercase h's, and he doesn't have it, so, but he only has 1 X, so I think it's 0%.
okay, so I'm looking to see if I can copy and paste really easily. No, that doesn't show it.
I was trying to draw a Punnett square on my hand, but I
Okay,
got
we
frustrated.
did that one. Did I skip one?
You did.
Yeah, okay. I was like, wait. All right. So if the woman has hemophilia, then she is, and I'm going to use the little arrow key. She's this. So the mom is where the arrow shows the superscript, right? She's that. I could be really technical about that. Well, let's see. That's going to be fun on PowerPoint, so I'm not going to bother. I usually copy and paste it in from Word after I do it. So the mom is, if she's X-linked and it's recessive and she has hemophilia, then she has to have both alleles be recessive to show it. So she has to be X little h, X little h, okay? And a man who does not. Now, since it's recessive, if the dad was this, then he would have hemophilia because he would have one X and no dominant X to block it out. So what do we know the dad has to be? He has to be this, okay? So the mom's gametes we put here, we'll put them in the top. And she only has one choice. She can only give an allele that codes for hemophilia. And the dad gives a happy X that doesn't code for hemophilia, or he codes for males, the Y. So all of the girls are going to get dad's good H and one of mom's not so good H's. So all of the girls are going to be what? They'll be heterozygotes, but will they have hemophilia? No, because they have one dominant allele. Each of the girls, any girls that they have, sorry, my mouse is not cooperating with me. Each of the girls has a version that codes for the protein that will allow blood to clot. And each of the sons, what are all of their sons going to end up with, thanks to mom? What do you see happening here? All of the sons will have what? Because they can only inherit a recessive allele. So what is the chance that each female child will have hemophilia? 0%. What is the chance that each male will have it, conversely? 100%. Okay, let's back up and do hypertrichosis, because I accidentally, did I skip more than one? And we did that one. This is the one that skipped. So this is subscripts. Hypertrichosis or hairy pinna. I'm sure you've seen dudes with hairy ears on the outside. It's a dominant trait found only in males. So if only males have it, what chromosome must it be on? It has to be sex linked. And if only males get it, what do females never get? A Y chromosome.
The Y.
So it has to be on the Y chromosome. And if it's dominant, do we use a capital letter or a lowercase? We use a lowercase. So only one of these has a Y in it. So it would have to be D. And whew, fortunately for us, this one shows the capital letter. So the answer is D. By the way, if you go back through here in the video, see how it says answer 4? That means it's D. And so the way I'm showing you, the answers are down here. Just don't cheat yourself. Try to figure it out before you look, okay?
You said a dominant trait is a capital letter, right?
Yeah, but X and Y are the sex chromosomes, and then it's this little H means recessive. This big H means dominant. And so it's here, this big H right here means hairy dominant ears. Okay, so this one isn't actually showing it right. It should be with the big eyes here. And I also meant to change it to letters. The way that this should be is with, it should be big I. It should be like this. And again, I'm using that a little caret key to show that it's a superscript. And this one should be, oh, that's wrong. Stop telling me my internet connection is unstable and just work for 5 more minutes, okay? That's what that one is. And this person, the correct way to show it, oh, it just messed it up. Is like that. And this person, the correct way to show it is okay. So the person who is B, O. What do we know that they are? We would write it like this. Which person can give blood to this type B blood person? Can they get type A blood? No, it'll be bad because they'll have an immune response to the A proteins or antibodies. So the only blood that they can get is the type O blood. Okay. So then here we go with homozygous purple and homozygous white flowered pea plants. So if they're homozygous purple and homozygous white, that tells you that they were both true breeding, okay? And their offspring have purple flowers. So the breeding of those individuals shows all purple flowers. So what does it demonstrate?
Dominance.
Dominance, that's right. Part of the reason Mendel avoided all of the characters that appeared to like merge together as opposed to being inherited separately was there was this idea that inheritance was blended, and he was trying to figure out if that was true or not, and so he was looking for evidence that the blending model was false. It is false, but it's hard to tease out when you look at things that are inherited on the same chromosome. Okay, number 19. This is getting at the 9 to 3 to 3 to 1 ratio again. The fact that all 7 of the pea plant traits studied by Mendel obeyed the principle of independent assortment most probably indicates which of the following. Do you think, how did he come up with the law of segregation if they didn't obey it? He specifically looked at 7 that did, so A is wrong. The diploid number of chromosomes in the pea plants was 7. Okay, well that's just trying to psych you out and get you to think that 7 traits means 7 chromosomes. They're not the same terms at all. So B is false. In fact, I don't even know how many chromosomes a pea plant has. I haven't looked it up in a while and I've forgotten. C, all of the chromosomes controlling the traits were located on the same chromosome. What do we know that he in fact did inadvertently by avoiding traits that travel together? He was looking specifically at traits that are genes on different chromosomes, which is D. And E is just absolutely false because it says gamete, the formation of gametes is by mitosis, and it's meiosis. Okay, so that's how you take that one apart. And the last one is, do you remember when there are 2 genes and one controls or gives a particular trait? So in this case, gene S controls the sharpness of spines in cacti. And cacti or cactuses with a dominant allele S have sharp spines, whereas homozygous recessive SS cacti have dull spines. And there's a second gene that determines whether cacti have spines at all. So what does it mean if you have multiple genes for a particular phenotype and one turns the other off or on? What do we call that?
It's B or D, but I can't remember which is which.
Pleiotropy is multiple phenotypic effects, like cystic fibrosis and sickle cell anemia.
We're going to go with epistasis.
So it's epistasis. Yep. Okay. So you will see questions like these, or maybe even these, on the test. Okay. I am trying to get my mouse to work again.